Hello everyone, welcome back to CAPSA. Today we will learn about LAL test. Let's quickly go through what we will learn today from this lecture. So what is LAL test? Where does blue blood comes from? What are the main testing methods? What exactly are endotoxins and why are they dangerous? Why can't we just use heat sterilization method to get rid of them? What is the difference between qualitative and quantitative LAL tests? What are the interfering factors and what are the alternative methods? Coming to the first, what is LAL test? LAL stands for Limulus Amebocyte Lysate. It actually detects and quantifies bacterial endotoxin. Detect and quantifies means which type of toxins are present and how much amount is present. Endotoxins come from mainly gram-negative bacteria. These toxins cause severe fever and shock in humans. So let's understand what does exotoxin and endotoxin means. Exotoxins are mainly produced inside the bacterial cell, but endotoxins are the lipopolysaccharide membrane or the virulence factor that is present, or the LPS factor present in the bacterial cell wall. So when the bacteria lysis or the cell damaged, this part or the lipopolysaccharide part comes out. Where does blue blood comes from? So blue blood of the Atlantic horseshoe crab. The Limulus polyphemus is the scientific name of this Atlantic horseshoe crab. The blood cells or the amebocyte actually clot when they touch the endotoxin. And the crabs are mainly collected. After collecting the blood, they are again returned to the ocean. Okay, so actually the blue color of the blood is due to presence of copper containing protein named hemocyanin. But the human blood is red, right? Because iron containing protein hemoglobin is present. Okay, so what are the main testing methods? Three type of testing methods are used: gel clot method, mainly laboratory method, preferred laboratory method, turbidimetric method, and chromogenic method. Okay, so gel clot method, simply yes or no testing, looking for a solid gel formation. So in this method, we just mix the sample with LAL reagent and incubate at 37 degrees Celsius. If endotoxins are present, gel will form, and when the tube is inverted, the gel remains intact. Okay, so if the gel remains intact, it will show positive test. You can see in the image here. And if the gel does not remain intact, then it is the negative test. In the turbidimetric method, we just measures the increase in the turbidity or the cloudiness over the time. So turbidimetric method is also known as kinetic turbidimetric assay or KTA, using a specialized turbidimeter. Okay, we just measure the turbidity or the cloudiness. Chromogenic method is used to measure a color change when the endotoxin trigger a dye. So for this method, we just use the sample, mix the sample with the LAL reagent, then incubate at 37 degrees Celsius. If the enzyme cleave the chromogenic substrate, what we are using gives the yellow color. Okay, and the instrument measures the absorbance, and this yellow color is due to the release of p-nitroaniline or para-nitroaniline. Okay, so this para-nitroaniline actually comes from the chromogenic substrate we are using. Okay, if the enzyme cleave that chromogenic substrate, it will release the p-nitroaniline and the yellow color will shown. So what exactly are endotoxins and why are they so much dangerous? Toxins are located within the outer membrane of the gram-negative bacterial cell. Very, very important point. You should remember this. Release when the bacteria multiply, or when the cell wall dies, or the simply burst. They are highly pyrogenic. It means it can trigger the intense fever. Pyro means fever, and can lead to septic shock, organ failure, death if injected. So why can't we just use heat sterilization to get rid of them? Endotoxins are exceptionally heat stable molecule. Okay, so standard autoclaving kills the live bacteria but leaves the endotoxin, because the standard autoclaving temperature, you can remember, is 121 degrees Celsius, but it requires extreme heat, typically over 250 degrees Celsius, for at least 30 minutes. Now, what is the difference between qualitative and quantitative LAL tests? So, for the qualitative method, we use gel clot method, the preferred laboratory method, that tells you if the endotoxins are present above a limit or not. For the quantitative assay, we use turbidimetric and chromogenic method to calculate the exact amount. What are the interfering factors in an LAL test? Substances like if there is protein, carbohydrates, chelators, detergents present in the sample, it will interact with the LAL reaction. If there is any chemical, it will stop the gel formation. If other chemicals like beta glucans are present, it can show the false positive clot. So you should remember this point, that the false positive clot is due to the presence of beta glucans. Now, are there any alternatives to using the horseshoe crab blood? Yes, there is a recombinant factor C assay. It is an engineered synthetic protein that mimics the crab's blood clotting enzyme. Benefit is eliminating the need for animal harvesting and ensures the sustainable supply chains. So that's all for today's lecture. Join me in the next lecture as we explore another interesting topic. Thank you.